award ceremony. I am Eveline van Rijswijk, I am a science communicator and I will be your moderator today, and I also hope to learn a lot today about cancer care and cancer research. Now, are you ready for the official welcome? Yeah? So we are here at the Royal Netherlands Academy of Arts and Sciences, and the Academy is also responsible for the process of nomination and selecting the prize winner of this Bob Pinedo Cancer Care Award. And therefore I would like to give the floor to the President of the Royal Netherlands Academy of Arts and Sciences, Marjolein Dochterom. Give her a warm hand.
Thank you very much, Eveline. So indeed, it's my huge pleasure to welcome all of you here in the room, but also online, on behalf of the Royal Netherlands Academy of Arts and Sciences. And as was already explained by Eveline, the Bob Pinedo Cancer Care Award is awarded every 2 years to an international acclaimed researcher in cancer care. It's not... No, it doesn't work? Let me see, I can keep talking slowly until it does work. Should I turn on any buttons? Is it working? Yes, now it works. Okay. I'm not going to start completely over, but welcome everybody on behalf of the Royal Netherlands Academy of Arts and Sciences to this award ceremony. Welcome to the laureate today, and also to Bob Pinedo, as was already introduced by Eveline. The award is offered by the Bob Pinedo Cancer Care Award Foundation, as was already mentioned, and since 2016, so this is now the 4th instance where the foundation has entrusted the selection of the award winner, Professor Zitvogel this year, to our academy. And the award winner receives a bronze sculpture in the form of Dr. Bob Pinedo's hand, which we will award later, symbolizing both strength and hope both strength and hope of healing, and very importantly also €100,000 that can be used for research. I would also like to mention here who was actually in the jury this year. This year the jury consists of Professor Pankras Hogendoor. He's the chair of the jury, and you will see him later in the video message that he has recorded. He's a professor of pathology, vice chair of the board of directors and dean at Leiden University. There's also Dr. Hanneke van Laarhoven. She's a professor of translational medical oncology at the University of Amsterdam. Dr. Henk Verheul, professor of medical oncology, Erasmus Medical Center, Rotterdam. And I've been told that you are in the audience. Very welcome to you as well. And last but not least, Dr. Wienette van der Graaf, professor of personalized and translational oncology at Radboud UMC, and also a group leader at the Netherlands Cancer Institute here in Amsterdam. So I want to also personally thank the jury very much for the important work that they do in selecting this very amazing laureate that we have this year. Just a few words about our academy, in case you are not familiar with our Academy of Sciences. We actually have 3 roles. We of course serve as a learned society, and representing in fact the full spectrum of scientific and scholarly disciplines. So humanities, the social sciences, medical sciences, and natural and technical sciences. We are also an advisor to our government on both science for policy and policy for science. And maybe not all of you are aware, and it's not the case for all academies around the world, but we also act as a management body for national research institutes in the life sciences and the social sciences and humanities. And the last thing to mention is we are also very proud to have under our wings, as you could say, a young academy. In 2005, we started the Jonge Academie. So this is a group of 50 early career scientists who serve a term of 5 years on this young academy, and they're very active and visible in the national debate on science and science policy. And they were one of the first internationally to be started, and you see now around the world that these young academies actually appear in many countries, and we are proud to say that our young academy has a very, serves as a role model for young academies worldwide. Since 2014, We also have a Society of Arts, the Academie van Kunsten. This is of course how it was intended in history, to have also arts under the wings of the Academy. And this is now the case again since, 8 years. I think that was enough information about the Academy. We're here, of course, to learn more about cancer care, the research of our laureate, and the symposium that we will have. So I would say enjoy the afternoon. And yeah, like Evelien was saying, let's make this a party. Thank you.
Thank you very much, Marjolein Dochterom. He was already mentioned in your speech. We will now hear great words about you, Laurence. This will be a laudatory speech given by the head of the jury, Pancras Hogendoorn. Enjoy it.
Ladies and gentlemen, many children dream about what they want to be. Police officer, doctor, professional footballer, nurse. However, not all of them make those dreams come true. But for some, the passion and will to achieve their goals are so great that they succeed. Laurence Zitvogel was one of such a child. She was only 8 years old when she decided to become a doctor and to develop a vaccine to fight cancer. Purposeful and with boundless energy and dedication, she has been working towards that goal ever since, with groundbreaking and hopeful results as a consequence, for both science and her patients. This makes her an outstanding laureate for the Bob Pinedo Cancer Care Award. This award is presented every 2 years to a scientist who is or has been particularly committed to scientific research on cancer and care for cancer patients. Laurence Zitvogel is full professor of immunology and biology at the University Paris-Saclay and scientific director of the clinical biome program at the Gustave Roussy Institute in Villejuif. She has devoted herself to research at the interface of oncology and immunology and immunotherapy since the early 90s at the University of Pittsburgh, United States, and then at Gustave Roussy. She has been a pioneer in the field of immune oncology research for introducing the gut microbiome into the complexity of anti-cancer immunosurveillance. She published papers on toll-like receptor 4 dependent contribution of the immune system to anti-cancer therapy, and also on the influence of the microbiome of the efficacy of PD-1-based immunotherapy for cancer and immunotherapy by CTLA-4 blockade. These papers are not only hallmarks for her scientific oeuvre, but also pivotal in current anti-cancer literature. She uncovered the immunosuppressive role of antibiotics in immunotherapy of cancer and the impact of the gut microbiota composition in regulating anti-tumor immunity during cyclophosphamide and immune checkpoint inhibitors therapies. Her work has been of the highest standard of scientific quality and has a promising future for the impact on cancer care. Thanks to her findings regarding the key role of the immune system in anti-cancer therapy efficacy, future treatments can be better adapted to the individual patient. In addition, it may be possible to improve the immune system so that it is better able to find tumors on its own, for example by administering certain microbiota. Laurence Zitvogel combines her dedication to research with a commitment to patient care. She also supervises a great authored or co-authored more than 500 publications and is one of the most cited researchers in her field. She has received various awards for her work and has been nominated as an officer in the French Ordre National de la Légion d'Honneur and at the French Academy of Medicine. She's also involved in integrating basic and clinical research groups and regularly organizes local and international meetings bringing together researchers in her field from across Europe and beyond. Ladies and gentlemen, Laurence Zitvogel is not only a gifted scientist, but also a dedicated doctor to her patients. Je ne supporte pas de perdre un de mes patients, she said in an interview. She is a role model for young scientists and the hub of an international network of researchers and research groups. She does all this with great energy and perseverance. C'est une tornade capable de déplacer des montagnes, is how she was characterized by one of her PhD students. It's likely that there are other 8-year-old children who would like to become doctors and who want to fight cancer. I hope they will fulfill their ambition with such much tenacity and passion as Laurence Zitvogel did and still does. Because of her inspiring dedication to cancer immunology research, as well as her impact on clinical care, the jury is unanimously convinced that Laurence Zitvogel is a worthy recipient of the Bob Pinedo Cancer Care Award 2022. Laurence, please accept my warmest congratulations on receiving this award, which consists of 100,000 euros for research and a sculpture in the shape of the hand of Bob Pinedo, the doctor and researcher after whom the award is named. The sculpture is a symbol of strength and hope for recovery. I hope this award will encourage you to continue your valuable work, for the sake of research, but above all, for the patients to whom you offer strength, hope, and prospects. I wish you every success.
Ooh. Really nice words for you, Laurence. Let's go to the official moment. Let's go to the award ceremony. May I invite to the front Marjolein Dochterom to hand over the award, and Douwe Breimer, board member of the Bob Pinedo Cancer Care Award Foundation. And may I ask Laurence Zitvogel to come to the front to accept the award and the sculpture. Let's give her a warm hand.
Very honored.
And this sculpture.
Thank you very much, everyone.
Let's do a photo moment. And may I also ask Bob Pinedo to join the award winner. And please give him a warm hand as well. Congratulations, Laurence. May I give the floor to you?
Thank you, Evelyn.
Yes.
It's going to be very difficult to start with such a beautiful, nice, and meaningful introduction. I am looking for the pointer. Thank you very much. Dear Commander and Professor Pinedo, dear academicians, dear award committee, dear collaborators, ladies and gentlemen, it's really an honor. I am very moved to be the lucky recipient of the Bob Pinedo Award 2022. I would like first to, of course, thanks Douwe Breimer, Pancras Ogendom, who did this beautiful introduction, Marjolein Dochterom, the Academy in general, the award committee, and all the philanthropists for their generosity towards medicine and research. So, as outlined by this introduction, my first thoughts are going to my patients. Actually, sorry about that. It's really tough for me to think about the ones that I never saved. I think a lot of us are thinking about them. but in fact this is the most important energy that pushes us forward and to find the lead to do progress and to continue our advances in medicine. So this statue is of course the hope for recovery, and I promise to them to spend my life with my colleagues, my team and you to make the best out of research. So I would like to thank my French institutions that supported me over the last decade, the 2 last decades. Gustave Roussy, the University of Paris-Saclay and INSERM, that were really supportive over the years. And of course, this distinction is shared by my most tedious and talented PhD trainees and postdocs. Some of them became very famous, as some of you know, and I am really sharing this distinction with these teams. I remember my old days at the University of Pittsburgh with my first mentor, Michael Lotz, a metastasis from the NCI, where Rosenberg and colleagues were the first to adoptively transfer T cells, and I was really mesmerized by this school of Pittsburgh. And I have, of course, a deep thinking towards our dearest Ralph Steinman, a Nobel Prize winner that died one day before receiving his prize. And I am also indebted to my first mentor in France, Wolf Hermann Friedman, that most of you know quite well. Of course, without the European collaborators that share their thoughts, their reagents, their nice energy to write guidelines of oncoimmunology prior to the burst of immunotherapy, I have, and I am so, so thankful. And I would like to today particularly thank Dr. Cornelius Melif, because he was one of these tumor immunology chauvinists. He was spearheaded the field of peptide-based cancer vaccines, and I remember my old times at the Keystone meetings when I was really completely absorbed by his inspiring talks. And also, the best director ever since of Gustave Roussy, Dr. Lex Egermont, who actually not only supported my work and that of my husband Guido Krömer, but also was one of the leader and the key opinion leader in melanoma research, in melanoma therapy, and has been always very positive about trying to get the best out of our research. Thank you very much, Lex, for all what you did to me, for me. So, of course, I would like to finish with my son and my husband. My son, Ulysses, I thank him for forgiving my absences, my heavy traveling, my work, alcoholism. But despite all of that, he majored at the University of Paris Medical School and he embraced the same career, so we didn't discourage him so much. And of course, last but not least, my husband, one of the brightest scientists that I ever met. Thank you, Guido, for this very relentless and insightful discussion that we had, and for supporting me over all the challenges that I had to face in these decades of career. And you know that if you don't have the support at home, it's much more complex. So France has been at the front line of infectious disease, of microbiology, of commensalism, historically with Pasteur, Metchnikov, Raoult, Doré and Ehrlich. And this Chinese bibliometric analysis is pointing to the fact that the gut microbiota now seems to earn its credential in the field of cancer immunotherapy, and suddenly my balls became bigger than Guido's balls, as you can see here on this graph, according to this Chinese. So the gut microbiome has now earned a spot in immuno-oncology, as recognized by the famous magazine Nature, that recognizes the gut microbiota now among the milestones in the oncological advances. And Hanahan has now added the gut microbiota and the tumor microbiota among the hallmarks of cancer. So this is due to a heavy work and a very cheerful and coordinated effort that was orchestrated around the world with all these pioneers of the gut microbiota immuno-oncology, and I have to thank them warmly for coordinating our dissemination of the results in an efficient way. And also to the bodies, the funding bodies, the European Council, the Ministry of French Health, and also the CIRE Foundation. So the field of oncology has been revolutionized by the advent of immune checkpoint inhibition. So these are antibodies targeting, monoclonal antibodies targeting inhibitory receptors expressed on the T lymphocytes. And these antibodies unleash T-cell functions, enabling T-cell dependent tumor regression and long-term benefit of immunotherapy to the patients. However, despite a lot, so at least 7 drugs that have been approved across 21 different indications, still some patients fail to respond, sadly, and this is because their immune system is stalled. And we have to get the question as to what governs the cancer immune set point, which is a very threshold beyond which an immune response can ensure in a given individual. And we believe that cancer genetics, host genetics, but also co-medication, comorbidities, environmental factors, and of course the microbiota play a critical role to set up the cancer immune system. So these are my lips, the microbes on my lips that are growing on the blood agar plate. However, I am not dangerous, and we have to acknowledge we are a meta-organism in which our mammalian part has co-evolved for millennia in a mutualistic symbiosis with a variety of different microbes, so bacteria, but also viruses, phages, archaea, parasites, and new guys that we nominate Candida phyla radiation. And this on a spread over 200 square meters of intestinal surface, separated by the inner milieu of only 20 micrometers, to exert seminal functions, eliminating the pathogens, tolerizing the food antigens, mediating the metabolism, ensuring the peristaltism, and more importantly, maintaining the intestinal barrier fitness and ensuring the maturation of the immune system for a perfect host fitness. So today I will try to go through 4 main pillars. What are the lines of evidence that support the impact of the gut microbiome in cancer immunotherapeutics? So the fact that antibiotics can be deleterious to their efficacy, also that the composition of the gut microbiota actually predicts resistance to immunotherapy, but also preclinical and clinical data showing that the gut composition, what we call the taxonomic composition of the gut microbes, are actually causatively linked with the efficacy of immunotherapy. So first, let's start with antibiotics. So in 2017, we took the risk on 175 patients, so it's not a lot, demonstrating, and I remember my discussion with Lex at that time, trying to take the risk to demonstrate that the 30% of individuals that had taken antibiotics were actually not so much benefiting from immune checkpoint inhibition, as you can see on these gray curves. And this was even independent of the other prognostic parameters. And it was on 170 patients that were non-small cell lung cancer in second-line therapies. But this has been rapidly extended to second-line immune checkpoint inhibition in the context of advanced kidney cancer patients. And once again, those that took antibiotics didn't go well. And now this has been extended to 12,000 individuals. 25% of them took antibiotic minus 60 days before or plus 30 days after the start of the first administration of anti-PD-1 or PD-L1 antibodies. And you can appreciate that when the antibiotics have been taken just prior to the first administration, the chances not to respond are by 2.3 times higher. And this is because the antibiotics are actually modulating the composition of the gut microbes with some guys that we now call the species belonging to Enterocluster genus are taking over at the expense of the beneficial bacteria that are named Lactospiraceae or Oscillospiraceae, for those of you who are familiar with these names. So the causality links between the composition of the microbes in the intestine and the likelihood to respond to an immunotherapy has been brought up bizarrely in what we call avatar mice, which are animals that are germ-free or antibiotic-treated to wipe out the endogenous flora. And then we adoptively transferred by oral gavage the fecal material from patients. We wait and establish a cancer and treat these animals. And so we could draw the conclusion very early on that when we were transferring fecal material from non-responders, we were conveying resistance to the animals to their immunotherapy, while in contrast, when we were gavaging the animals with fecal material from responders, then we would confer sensitivity to these mice. So it meant that we need a tool, a diagnosis tool, to align the composition of the gut microbiota, and so what we would call a gut dysbiosis. And to achieve this goal, we built up onto a network in France made of key opinion leaders in lung cancers that collected more than 1,000 fecal material from their patients prior to the start of immunotherapy. And thanks to bioinformaticians that perform what we call shotgun-based metagenomics analysis, and relying on random forest-based machine learning, we could work out a program, an algorithm that could predict resistance or a sensitivity to a given patient to immunotherapy. And so we leveraged this French network to a European dimension by winning this European Council program, gathering 17 partners in 8 countries. And today, Yolanda De Vries is among us, and she joins this network that aims at probing the geographical locations, the lifestyles, and also the diet habits in the modulation of the gut dysbiosis in these patients to ameliorate our diagnosis tools and also propose prophylactic measures to re-establish responsiveness, to restore responsiveness. So a third extension beyond Europe was Israel and was United States, and this was thanks to the CIRA Foundation that we could achieve such a broad network, and this allowed us to perform a metagenomics analysis now up to 9,000 cancer patients. So today I'm not going to show you the results of the 9,000, but just enough to tell you that out of more than 1,500 cancer patients for whom we established the metagenomics composition of the stools, comparing it to healthy volunteers-derived poops, we ended up showing in red that the cancer patients have a different composition than the non-cancer patients, the healthy volunteers, with an overdominance of microbes that we will call harmful microbes that are mostly belonging to the Enterocluster genus. And these microbes in red here are the same as those that we identified in non-responders. And that's always the same, the species from the genus Enterocluster, and they outrule those that are actually associated with an healthy status, the Lactospiraceae and the Oxylispiraceae belonging bacteria. So in other words, cancer patients show up with a gut dysbiosis that is not specific of the chronic inflammation of cancer, but is actually pathognomonic of any chronic inflammatory disorders that we can find in cardiovascular disease, individuals in mental disease, in gastrointestinal inflammatory pathologies and so on. So, in other words, the gut is somehow sensing the cancer located at a distant site. And in fact, that's what we demonstrated in mouse models, that the ileum is the organ, is the first brain. The ileum is sensing a neoplastic process expanding at a distant site from the intestine. And so the ileum senses the cancer. This is named stress ileopathy because this sensing results from a disbalance between parasympathetic and sympathetic signaling. And this is not neutral because this stress ileopathy resulting from cancer progression is actually participating to the incidence and the severity of the cancer through this protracted dysbiosis. So in fact, just to show you one piece, first of all, this graph is very beautiful, I believe, very colorful. But this graph is actually illustrating that when cancer is inoculated, the ileum becomes atrophic, and the patchy atrophy of the ileum, not the colon, just the ileum, is prototypically defined as the apoptosis of the crypt, the proliferation of the ectopic proliferation of neuroendocrine cells, the degranulation of the Paneth cells, the proliferation of the goblet cells, the vasoconstriction of the villus vessels, the permeability of the gut that is transiently increased, giving rise and paving the way to the overgrowth of harmful bacteria. And this is actually caused by a disbalance between cholinergic and adrenergic signaling in the gut. That's why it's a stress ileopathy. Once again, the cancer-associated stress ileopathy is not specific of cancer. It has already been described in the context of stroke, for instance. So to try to develop a more friendly user test, we focused on a bacterium that is very regulatory, the Akkermansia mucidifila, also well known from Patrice Cany and colleagues in Belgium, that describe at first this bacterium as preventing insulinoresistance. And in actual fact, we stumble upon the discovery that this bacterium is indeed, once again, associated with good responders when it is present. So in actual fact, only 30% of second-line non-small cell lung cancer patients do have residency in their intestine of this Akkermansia mucidifila, and these will be the lucky individuals that will benefit from PD-1 blockade. In sharp contrast, those patients in red that do not have intestinal residency of Akkermansia or have an overgrowth of Akkermansia in the context of a cancer ileopathy, those will have a dismal prognosis. So we could mimic this and try to show a causality link between Akkermansia residency and good performance to immunotherapy using this avatar mouse system, receiving by oral gavage the poops from Akkermansia positive or negative patients. And you can appreciate that those patients that have Akkermansia positive poops are readily responding to PD-1 blockade, while the ones that have not received, that have received poops that are Akkermansia negative will not respond unless we compensate with an oral capsule of lyophilized Akkermansia mucidifila. So this paved the way to the personalized therapy using a diagnosis test before you compensate the individuals that lack the good bacteria. So, what are the mechanisms of action? How come that the gut microbes can actually interfere in the reprogramming of the tumor microenvironment? So, basically, there are 4 pillars. They can work through stimulation of the immune system, through metabolic cues. They can directly act on the endothelial or the epithelial barriers. And so I will just summarize some of the vignettes. For instance, at the level of the immune system, we know that the microbes are, of course, presenting or harboring ligands of pattern recognition receptors to activate the innate and the cognate immune responses. But they also harbor in their genome some sequences that share up to 80% sequence homology with oncogenes. And so when T cells are primed, for instance, by sequences in a phage that lysogenize the bacteria, then these T cells will cross-react with the oncogenes overexpressed in a cancer. So this is also very interesting to note that the high endothelial venules of mesenteric lymph node, of Peyer's patches, of venules of the lamina propria of this molecule, the mucosal matcam. And this mucosal matcam is like a scotch in the intestine, retaining these alpha-4 beta-7 regulatory T cells that normally keep in check the gut from the autoimmunity and the autodestruction of the epithelial cells because of the pathogens and because of the food antigens. And once this barrier is disrupted, the regulatory T cells actually leave the gut and end up in the tumor bed, promoting tumor outgrowth. So we also showed that the apoptosis of the ileal crypt, together with the ileal microbes, are actually 2 conditions to elicit a follicular T helper cell response that will be instrumental in the context of colon cancer. So I have to tell you that our organs are not sterile. What we learned in our textbook is more or less not really updated. The tumors contain up to 2 to 3 or 4 different species of bacteria and sometimes even fungi, as outlined by Ravi Strossmad at the Weizmann Institute. And these bugs, when they are invading the cancers and when they are infecting the tumor cells, they can promote inflammation. They can stimulate or trigger a sustained proliferative signaling. They will also generate avoidance of immunosurveillance. And of course, it has been noted that they can directly generate genome instability and DNA breaks. So they can be, of course, detrimental. But in some instances, they could be beneficial when this infection is actually ending up in antigen presentation at the level of MHC class 1 and class 2 of the tumor cell to expand T lymphocyte. So this was an interesting concept that has been coined by the Weizmann Institute at the first place, by Yardenia Samuels. But we find out that it is actually true in bladder tumors because the bladder cancers is actually infected by E. coli, the Escherichia coli, what we call the uropathogenic E. coli. And this uropathogenic E. coli is actually very immunogenic. And so the patients that have seen this bacteria and that have been infected in the past have mounted follicular T helper cell responses as well as B cell responses that can be considered as memory T cells and B cell responses. And this is quite nice because those T cell responses can be reactivated, reinvigorated by neoadjuvant pembrolizumab in the context of this muscle invasive bladder tumors. So the scenario that we have in mind is the following one, that patients have residency of Escherichia coli in their intestine. These will translocate at some point and infect when those cells have colibactin or so on, or other virulence factors. They can translocate and directly infect the mucosal area of the genitourinary tract. This will help mounting T and B cell response and tertiary lymphoid structures that will, once established, be reactivated by pembrolizumab and will amplify the maturation of the tertiary lymphoid organs and really orchestrate the immune response locally so that the patients get to respond. And in actual fact, it's very easy to look at the IgG titers, which are a proxy of memory B cell responses against E. coli. And so the IgG titers against E. coli are actually best predicting the response to pembrolizumab in these muscle-invasive bladder tumors. This is the first study, the second study, and the first study. This is a Dutch study, actually. These are stage 3, this is a stage 4 study, and now this is another stage 4 study, the Yopredi AstraZeneca phase 3 trials using durvalumab in the end-stage bladder tumors, metastatic bladder tumors, and you see that once again the IgG response against E. coli seems to be a proxy of memory responses directed against pathogens infecting the tumor. So, of course, you will tell me maybe you were lucky and you just stumbled upon E. coli, but we have tested up to 15 different local pathobionts, and only E. coli was actually standing out as immunogenic in this context, not the other bacteria. So, now let's come to the very notion of how to harness the gut microbiota to launch new therapeutic interventions and avenues. I will quickly go through the 4 different quadrants here. So either exploiting complex immune ecosystems, gut immune systems, so fecal microbial transplantation from healthy donors or cured patients, and we will hear the beautiful talk from Hanen's lab. We can also think about prebiotics, which are small components, metabolites that can be utilized by the gut microbes to amplify their prevalence or their function. Probiotics, which actually means live biotherapeutics, and some of them are already in the clinic, and diets, either fasting, fiber-enriched, or ketogenic diets are currently used. So I will quickly summarize these 2 pilot studies that are quite fascinating, one from the Sheba Institute in Israel, the other one from the University of Pittsburgh, where the investigators targeted the advanced melanoma patients that were primarily resistant to PD-1 blockade, and they changed their microbiome by transferring, by encapsulated lyophilized complex ecosystems, allogenic materials from cured patients, melanoma that were cured or in very prolonged partial response. And they could get, in 1/3 cases, they could circumvent the primary resistance by reintroduction of PD-1 blockade. And this was by reprogramming not only the fitness of the intestinal barrier, but also by promoting an activation of the systemic immune tonus, reducing the systemic inflammatory markers such as IL-8, and modulating the metabolome. So this second study is quite compelling. This is a study from San Francisco, from Montipal, that tried to ameliorate the response rate in first-line advanced kidney cancer patients treated with ipilimumab, together with the Clostridium butyricum, and it could really significantly improve overall survival in these patients. diet, either ketogenic diet by boosting the T cells and preventing the PD-L1 upregulation despite high levels of interferon gamma. Ketogenic diet and its ketone body, the 3-deoxybutyrate, are key in launching and synergizing with PD-1 blockade. And this very nice story from the MD Anderson showing that high fiber diet, when in the absence of probiotic, is capable of boosting the results that we can obtain from PD-1 blockade in advanced melanoma patients. Last but not least, a very simple way, the camu, which is a Brazilian berry that contains this polyphenol castalagine. So Bertrand Routy was capable of showing that camu can do better than PD-1 alone, and that's because camu was able to shift the microbiome composition, enriching for Akkermansia and Ruminococcus species. And in fact, this is because the Ruminococcus are very happy because they receive their food every day, the castalagine from the berry, because the fluorescent castalagine is labeling the Ruminococcus species residing in the poops, and this maintain the necessary equilibrium for the beneficial species against the harmful ones. So finally, what are the implementations for the future? I believe we should first target the patients that do not respond quite well to the first-line immunotherapy. But before we think about labeling them dysbiotic, we need to have diagnosis tools to make sure that the resistance is due to a gut dysbiosis. So diagnosis tools are important, as well as the anamnesis of the patients, if he took or not proton pump inhibitors, laxative or antibiotics, and then trying to adapt the the treatment, the microbiota-centered intervention to the gut dysbiosis according to different modalities that I just summarized to you earlier. And use all the tools we have, so immune response against the microbiota, but also metabolomics and the shift in the gut composition will be all important to follow up the efficacy of our microbiota-centered interventions. And that's what we try to do at the Clinicobiome together with Lisa De Rosa. We are opening now, it's already open. We have opened the Keto-Rhin, so ketogenic diet and 3-deoxybutyrate in advanced first-line kidney cancer patients, as well as Akkermansia species in lyophilized encapsulated formulation developed by Everimmune in kidney and lung cancer patients. And we are about to open fecal microbial transplantation for antibiotic-treated individuals. So with that, I will close, and thanks again the team, the CIRA Foundation, that is supporting these clinical trials. I wanted to say that the merit is going to Lisa De Rosa and Bertrand Routy, my two young PI that really did all and led all these works together with the rest of the team. But I think this award, this wonderful Bob Pinedo Award, will be the 100,000 euros will be utilized for all these clinical trials to try to improve the response rate in our patients. And I would like to, of course, thank all the other members of my team that are beyond, but really in the front line of culturomics, immunomics, metabolomics, inflammomics, proteomics, and so on, and metagenomics. And the bioinformaticians, the wonderful Nicolas Segata, Valerio Yeba, Andrew Thomas, the INRAE collaborators, as well as Guido Krömer, my husband, for the metabolomics declination, and Didier Raoult for the culturomics, my former postdocs, the key opinion leaders, the network in France for kidney and lung cancer patients, and our new postdoc, Emmanuel Macron.
Very much, Laurence Zitvogel. I open the floor for questions. I can imagine you have a lot of questions, and of course, Bob Pinedo, the first question is for you.
Fascinating, Laurence. I think the tumor microbiome is very important and plays an important role. Do you think that the colon cancer has more problems with the tumor microbiome than the other cancers? And could that be a reason why they are mainly resistant to immunotherapy? And have you ever considered to expose them, the primary, to the right microbiome?
This is an excellent question that would deserve almost another talk, Bob, because we are, of course, actively working on this paradox that the colon cancer is the first to have an immunoscore and the last to respond to immune checkpoint inhibition, except the MSI high, the Lynch in neoadjuvant settings. So, in fact, you're right. The colon cancers, the MSI, so the microsatellite unstable ones, have a different microbiota than the others. The Lynch have different E. coli, for instance, than the non-Lynch microbiota. So everything is different because of their genomic instability. I think the microbes in the gut are seeing the stress of the mucosa on a regular basis, and so change their metabolomic, they change their genotypes. And we are actively working on that, and seems the result seems to indicate that these microbes are more immunogenic, but they are also invading, and so they are the F nucleatum, the treponema, and so on. They are on one hand invading the dormant cells, as shown by Susan Bullman recently in the recent Nature. So the bad bacteria are actually causing the dormant cells to become, to undergo epithelio-mesenchymal transition and dissemination, are also involved in recruitment of neutrophils and exclusion of the T cells from the microenvironment. But these bugs are actually quite immunogenic, and I think the reason why they take over is presumably because the other part of the microbiota is stalled as well, and presumably because these patients have not mounted an immune response at the first place. So we are trying to understand that a little bit more, but I guess this is exactly at the center of the conundrum that still needs to be unveiled.
Another question.
Yeah, you must have thought about the interaction between immunogenic cell death caused by chemotherapeutics and what chemotherapeutics do to microbe invasion in the gut, yeah, and the microbiota. So, yes, because there are, I'm sure, deleterious and favorable interactions possible.
Exactly. So we published that, actually, Paola Roberti paper in Nature Medicine, August 2020. We published this result. So actually, oxaliplatinum is promoting immunogenic cell death of the crypt of the ileum. This gives rise to new bacteria that put up with this crisis. And the two together elicit follicular T helper cells, thanks to the mobilization of double positive lamina propria derived dendritic cells that can secrete IL-12 and IL-1 beta. So if you have this scenario, you elicit TFH, and if you don't have this scenario, you elicit Th17 pro-angiogenic T cells. So it is the genus face of the coin. So immunogenic cell death with the right bacteria, success for TFH. In the absence of that, the same chemotherapy would lead to Th17 priming instead.
We have room for one more question. Yep.
Koos van Hoeven, I'm a medical oncologist. First of all, congratulations with the Bob Pinedo Award, and thank you for your beautiful lecture about excellent work. I have a question from a patient perspective, because patients always want to do something themselves in lifestyle or diet to improve their outcomes. And can you give our patients who undergo immunotherapy an advice in a diet? So do they have to take a high fiber diet, or do they have to take probiotics, or is it too early and is everything work in progress?
Yeah, that's of course a question that is coming all the time up in the discussion. So at the moment, the easy answer is the last proposal, so it's too early to make any recommendation in the absence of clinical trials. Diet interventions have always been like, you know, an alchemy, so it needs some criteria and some biomarkers. You know, when we take, for instance, ketogenic diet, what do we mean by ketogenic diet? Can we monitor free oxybutyrate and acetoacetate? The pharmacokinetics of this ketone body seems to be important for the outcome, so that's one example. For the fibers and rich diet, it's the same. Some people are already taking fibers more than 20 grams, so those they presumably need a higher dosing to get the short-chain fatty acid at the right monitoring. And maybe we need a proxy. So if we had a proxy like a score, a diagnosis score, we call that the topo score in my lab. So a score that would enable the clinician to know whether the patient is really completely dysbiotic or more or less quite in equilibrium. It's also a different prospect for the advice. So I guess there are many parameters and it's not simple. One size will not fit all.
You mentioned some possible treatments on your slide. Pun intended, what is your gut feeling? What will help the most beneficial for the patients?
Thank you for playing with the words.
You're welcome.
Well, as I said, I think it's going to be more or less, well, it's not just that the personalization of treatment is always the answer, but in this particular context, I think that it depends on the dysbiosis. If you have an antibiotherapy that has been really reshuffling entirely the immune system and the gut composition, you would need an FMT, so a complete reconstitution with a brown new and completely fit gut ecosystem, so a fecal microbial transplantation. In contrast, if you just, you know, let's say, have a deficiency in some regulatory bacteria that are very critical, then we could envision to just advise an encapsulation of one single species.
I want to ask you to give a warm hand. Laurence Zitvogel. Well, Laurence, there are some more people here in the audience that want to congratulate you and who also want to share their knowledge with all of us. So that's what we'll be doing in the second half of this meeting, because I already said it today is about celebrating and about sharing knowledge. So the first two speakers I will introduce are Alexander Eggermont or Lex Eggermont and Jessica Borgers. Lex Eggermont is a surgical oncologist and professor of immunotherapy at the University Medical Center Utrecht and chief scientific officer at the Prinses Maxima Center. And his talk will be about you, Laurence. And then Jessica Borgers, she's a PhD student at the Netherlands Cancer Institute, and she will talk about feces microbiota transplantation to convert non-response to immune checkpoint inhibition in melanoma. Give them already a warm hand, and then they'll give their presentations. Lex Eggermont.
So, good afternoon, everybody. Laurence, what a brilliant moment. So I actually stepped out of the guidelines that I received regarding this session and what I was supposed to do. And this is not the first time in my life that I do that, but I have a very special reason, because it's all about Laurence Zitvogel. And I think that bringing my science slides on adjuvant trials with anti-CTLA-4 and anti-PD-1 in melanoma, and the current project that we are running on an immune globulin signature of different types of microbiome that actually are of tremendous prognostic potential, and even potentially of predictive potential, would take away from the focus on the question, how extraordinary is Laurence Zitvogel? And I think that merits just not another laudatio, but just some insight in some of the most extraordinary aspects of a person who is so unique. So basically, Laurence, you are the Maria Callas of tumor immunology. And you are the Maria Callas because of the total package, Laurence Zitvogel. So the first time we met, you were actually in the lab of Mike Lodes at the University Medical Center of Pittsburgh. And Mike Lodes was the first lieutenant at the time when I was in Steve Rosenberg's lab at the NCI in Bethesda. And Mike Lodes was known for his in-depth knowledge of the literature in immunology. And so we actually shared a journal club session in Pittsburgh, and the topic was an article in Journal of Immunology about HMGB, which was very novel at the time. It's one of those damp molecules that lead to immunogenic cell death. And so Mike Lodes took the spotlight, and he gave a short summary of 3 articles that might be, you know, commenting or in contrast to this article, etc., etc. Then Laurence was there, and she was wearing a neutral outfit like today in the United States in a lab, right? And she basically in 7 minutes gave a summary of whatever was published on this topic over the last years, and summarized not only all the different hypotheses, but suggested new hypotheses, split off hypotheses, how that should be investigated, and what was actually then missing in this paper, and why the hell this paper was chosen for a journal club. And you go like, so wait a minute, there is not even a shadow anymore where you can hide, because Laurence was the spotlight. So you are fireworks in many, many, many ways, and not just scientifically, but in everything you do. Also the way you teach your PhD students. So if there is a second word to describe intensity, it still needs to be invented, because you need to actually see it, how that works. So Laurence, always, working in a compressed time capsule, will not grill, but will instruct PhD students so efficaciously, with so many aspects, that all their receptors will be occupied within 1 to 2 hours for at least 3 months, and potential hypotheses to what, where, and with whom to address. And so you were 2 floors behind my office at Gustave Roussy. And you know very well that you and Guido were one of the reasons why I came to Gustave Roussy. And it's been simply extraordinary how we could develop an immunotherapy development program with you as scientific director. We recruited Aurélien Marabel from Stanford out of Ron Levi's lab as clinical director. And we went in no time from 1 phase 1 immunotherapy development study to 70 concurrently running phase 1 immunotherapy development studies. And we had another 220 running as later studies in the Institute. So it became by a mile the largest program in Europe. And that can only be done if there is priming and reverse priming. And we've had a relationship where there was priming, and then I tried to stay alive, and I tried the model of reverse priming, which she doesn't need, because she was already at another level of priming her environment and the whole institute. And so it's interesting when you look at musicians and the Maria Callas phenomenon. I also must say a word about Guido. Because, well, Guido, you're a very courageous man. When you are the partner of Laurence Zitvogel, you don't get a lot of attention. And it is not because she doesn't give you the space for that attention, but you know, this is the biggest walking sponge on earth that just absorbs not only all attention, but gives back immediately all explosive fireworks, counteractions. So you went on to become the most prolific writer in science that I know. And so when I think about you, because I can write very well. Both my parents were teachers, and they taught me how to talk and how to write. And so I write with ease. But you make me feel, you all know the movie Amadeus, about Mozart, right? So Guido, you make me feel like Salieri. And you are the Mozart. So we have here, Maria Callas, we have here Mozart. And you have been the diamonds of my almost 10 years in Paris. And fortunately, and that's my fortune and my gratitude, not only in the science sector, but also in our personal relationships, and you have become such good friends. And so it's with such pleasure that I'm here and standing in front of you. And Laurence, basically you are like AAA. What AAA, anybody who has been in the United States knows it's the American Automobile Association. It's the biggest association in the United States. They also have an NRA, let's not talk about it. But AAA for me is like Laurence, because for me it's admiration. For me it's adoration. And for me it's also je t'aime, because that also starts with an A. And so I thought that l'Académie royale des sciences aux Pays-Bas était exactement la plateforme qu'il me fallait pour cette déclaration publique de mon admiration pour toi, mon amitié et le fait que je t'aime. Bon, dat was wat ik wilde zeggen en daar zitten ze. Merci.
Totally different talk, but my talk will be about how we can use fecal microbiota transplantations, or FMTs, to help patients with melanoma. I have no disclosures. So as many of you know, immune checkpoint inhibitors have significantly improved the survival rates of patients with many different cancer types, including patients with melanoma. However, not all patients benefit from this, or some patients only benefit in the short run and not in the long run. So therefore, many attempts have been made to identify new mechanisms, and many of them are still ongoing, to see if we can also help the patients that do not benefit. And one of these mechanisms, thanks to the work of many people, but definitely including Professor Zitvogel, might be the gastrointestinal microbiome. The first evidence that this might play a role in anti-tumor immunity came from a mouse study. The study was published in 2015, and what they did is they had two genetically similar mice, but they came from two different vendors with known differences in their microbiome. And when they gave these mice similar melanoma tumors, what you can see over here is that one of the groups from one vendor had smaller tumors than the mice from the other vendor. This was, of course, very interesting, especially because they knew they had differences in their microbiome. So when they then transferred features from the mice with lower tumors to the mice with the larger tumors, they were able to convert this. Then, 3 years later, 3 papers were published in Science, and they took what they found in these mice studies to patients. So they did an observational study where they looked to see if they could identify parts of the microbiome from patients who were treated with immunotherapy with either response or with non-response. And as you can see in these pictures, they identified many different components of the microbiome. But if we look in slightly more detail, it's not super clear on this slide. You can see that for each study, they identified components that were associated with response and with non-response, and then for each of the 3 studies. And what you can see is that not a whole lot of overlap occurred. And also some were associated with response in one study, with non-response in the other study, and there was only one that actually overlapped between these two. So despite the fact that they weren't able to identify specific components of the microbiome, when they transferred features from these patients to mice and then gave these mice tumors, they saw when the patient was a responder, the mice also had lower tumor volumes, which was of course very interesting to see. This formed the basis of the first two phase 1 clinical trials where they gave melanoma patients FMT. As Professor Zitvogel already mentioned, the donors were melanoma patients that responded very well to treatment. And they gave these FMTs to patients that were not responsive to their immunotherapy treatment. These were the first trials in patients, and so the primary endpoint of these studies were, is it safe to give these FMTs to patients? And the answer is yes, it was indeed safe to give this. But even more excitingly, what you can see over here is the tumor volume of a patient prior to the FMT, same over here. And minus means that the tumor volume goes down. So in 3 patients in this study and another 3 in this study, the tumor volume went down after the FMT, while beforehand they were progressive. It's also nicely illustrated here. You can see melanoma metastases on the left leg, and after FMT they disappear, and this patient is in complete remission now. So that is, of course, really good news. Oh, and there was also here not a clear association between the composition of the FMT and the response of the patient. But there are still, because there's no clear association found, there are still many questions left to be answered. So for instance, what are the therapeutically relevant bacterial taxa? What is the optimal composition of an FMT? Which donors should we use? Should we use donors that are patients that are in long-term durable remission? Should we use patients that are responding right now? Should we use healthy donors? And for the patients that receive the FMTs, how should we administer the FMT? Should it be gastroscopy? Should it be colonoscopy? Should it be oral capsules? Should it be a single dose? Should it be a maintenance treatment? Should we pre-treat these patients with antibiotics because it's thought to enhance the engraftment of the FMT? Or shouldn't we, because there is also a negative association between the use of antibiotics and immunotherapy outcomes? And which patients will benefit most from an FMT? Well, luckily, there are currently 13 trials ongoing testing FMTs in an oncological setting. The majority of them is for melanoma patients, but there are also patients that can enroll with renal cell carcinoma, lung cancer, prostate cancer, pancreatic cancer, and they occur in sites all over the world. One of these trials takes place in the Netherlands at the Netherlands Cancer Institute, which I will tell you a little bit more about in the next few slides. So the trial at our site is an investigator-initiated study in collaboration with the Dutch donor Features Bank. And in our study, we want to see if we can convert the immunotherapy non-response of 24 melanoma patients with an FMT. But what is a little different about this study is that half the patients will receive an FMT from one of the 2 donors that are melanoma patients that respond to immunotherapy, and 12 of them will receive an FMT from a non-responding donor, because we want to see whether simply a change in the microbiome is enough to convert this immunotherapy response, or if it really needs to be from a responder patient as a donor. This is something that's not known yet. So what does it look like for the patients that participate in this trial? As you can see over here, we're looking for melanoma patients that show progressive disease on a CT scan, so they do not respond to their current standard of care first-line treatment, anti-PD-1. Then these patients will be screened, and if they're eligible, they will be randomized. So 12 patients will receive an FMT from a responding donor and 12 from a non-responding donor. And this is double-blind, so both the patients as well as the doctors do not know which patient or which FMT the patient received until all 24 patients have received their FMT. Then patients will undergo bowel preparation, first with antibiotics and then with bowel clearance, because it's thought to enhance the engraftment of the FMT. Patients will receive their FMT via gastroscopy and then will be followed with CT scans, all the while they continue with their anti-PD-1 treatment. Both prior to as well as after the FMT, we will collect blood samples, fecal samples, and biopsies from these patients. The endpoints of the study, the primary endpoint will be the efficacy. So can we convert this non-response to immunotherapy, which is defined as clinical benefits of patients that experience stable disease, a partial response, or a complete response. Secondary endpoint will be safety, and the study will be considered safe if less than 7 patients experience a severe toxicity. And there are several exploratory endpoints, for instance, what do we see in the gut microbiome, and this will be done in Leiden. And at the NKI, we will look at the blood samples and the tumor biopsies from these patients to look at the immune cell populations, both systemically as well as in the tumor microenvironment. And we want to correlate this with the efficacy of the treatment. The current status of the study, there are 2 responding donors have completed the features donation. So we have the 12 responding FMTs, but we're still looking for the non-responding patients to donate. Once all FMTs have been generated, patient enrollment will start, and we expect the results in approximately 2 years. With that, I would like to thank everybody involved, both at the NKI and in Leiden at the Dutch Donor Features Bank. And last but not least, congratulations, Professor Zitvogel.
Thank you, Jessica. Please try to remind the questions you have, because I'll introduce the last 2 speakers and then we'll have a question round for all the speakers. 2 wonderful people who also want to congratulate you and share their knowledge with you are Kees Melief and Marieke Franse. Kees Melief is Emeritus Professor of Leiden University Medical Center and Chief Scientific Officer at ISA Pharmaceuticals. And Marieke Franse is a Senior Postdoc Researcher at Translational Immuno-Oncology at the Amsterdam UMC. Give them a warm hand and enjoy the knowledge they'll be sharing.
It's really great to be here, Laurence. I congratulate you from the bottom of my heart. And we've had many interactions over the years at many meetings, dinners, whatever. It was always exciting, scientifically exciting and wonderful and productive. And in fact, you are a prodigy not only in the microbiota, but also in 2 subjects that have not even been touched upon, which I would like to say a few words about. And the first of these is exosomes. And I had the good fortune to run into exosomes when Graça Raposo was a postdoc in Utrecht in the laboratory of Hans Geuze. And we, in a very old car, we went every 2 weeks to Utrecht to talk about this, what these particles were. And what in fact they are, as you can see, they are produced from so-called endosomes, which then mature and can either go on to become lysosomes, or they can produce these particles. And we now know that all cells in the body produce these particles. And we were fortunate enough to show that these particles had antigen-presenting capacity. And, but Laurence immediately, thinking about patients, picked this up and decided that this could be a wonderful vaccine for patients. So she started to produce exosomes from dendritic cells called dexosomes. And you should know that the composition of exosomes depends very much on the cell that produces them. So tumor cells produce different exosomes than dendritic cells, and in fact the exosomes from tumor cells are likely very immunosuppressive. And people are now using exosomes from tumor patients for diagnostic purposes to identify them, what their composition is, and so on. So, and Laurence really did heroic efforts with these dexosomes. And perhaps, you know, she was way ahead of her time because the regulatory authorities were not ready to use dexosomes for cancer therapy, I guess. We should one day return to them. The other thing is, of course, immunogenic cell death, which Laurence already mentioned. And this is from one of her reviews, where you see that dying tumor cells upon treatment with certain chemotherapeutics produce certain molecules such as HMGB1, which then makes them immunostimulatory. So paradoxically, chemotherapy can be immunostimulatory. And we also had an interest in this because we have an interest in cancer vaccines. And there you can see that cancer vaccines can be either DNA, RNA, synthetic long peptides, or viruses, which upon injection subcutaneously or intradermally or intramuscularly, then elicit an immune response when they get to dendritic cells in the draining lymph nodes. And then they stimulate a T cell response, and these T cells, if properly stimulated through the thoracic duct, get into the bloodstream. And the great thing about T cells is that they seek out the tumor cells wherever they are. So it's a systemic treatment by, again, a local immune stimulation, like the microbiota in the gut. And so cancer vaccines have the potential to be strongly anti-tumor reactive. But the problem is that in a tumor microenvironment, very often there are immunosuppressive circumstances, like immunosuppressive enzymes or immunosuppressive myeloid cells or regulatory T cells or immunosuppressive cytokines and chemokines. And this becomes worse as the tumor becomes larger. And so what we have to do is a combination treatment to reverse this. And one combination treatment that you can do is chemotherapy. And here we see that chemotherapy alone, which is the light green line, didn't do much for this particular mouse tumor, which happens to be a human papillomavirus positive mouse tumor. And also peptide vaccine alone is not very effective, but together it's very effective curing 80% of the mice with established tumors. And so we were interested in Laurence's work also from this point of view. Is this immunogenic cell death, or is it something else, or a combination? And we used cisplatin, which in the hands of Laurence is not causing immunogenic cell death. So what was going on? So oxaliplatin was, and so what we showed, actually, what the combination did is in the untreated tumors, you don't see any T cells entering, but in the treated tumors, you see the peptide entering. And the cisplatin alone did not cause T cell trafficking to the tumor, but the vaccine did, but only together they were effective, as you can see. And the little graph on the right shows you that these T cells were also highly tumor-specific, HPV-16 specific. So this is what cancer vaccines can do. They can make barren tumors that have no T cells. They can make the T cells go into the tumor and mediate a therapeutic effect together with chemotherapy. And so what, to cut it short, a long story short, but without vaccination, chemotherapy works by depletion of immunosuppressive myeloid cells and by immunogenic cell death if you have the right chemotherapy. And with vaccination, you have the same two effects, plus synergy of cancer cell apoptosis between TNF-alpha release from vaccine-induced T cells in the tumor bed, and then the vaccine has made the T cells go into the tumor. And so we could probably, the reason why we did not use actually oxaliplatin, Laurence, was that the regulatory authorities wouldn't allow it, because for these tumors it was only carboplatin or cisplatin. And so that's, but hopefully also we would like to lower the dose of chemotherapy for the benefit of the patient, Because if you treat the immune system, the myeloid cell depletion, with chemotherapy, you can use a much lower dose than if you treat the tumor. Anyway, so this clinically this seems to work also, because those patients in combination with chemotherapy that have a strong HPV-specific response following combination of vaccination and chemotherapy, they are in the blue line, and they live significantly longer than those that have a low response to HPV-16. And this is not just due to immunocompetence, because the reaction to common microbial antigen in the two groups is the same. So it's specifically due to the vaccine-induced response against the oncogenic HPV-16 virus. Now, and we know that you need both CD4 and CD8 T cells to achieve this in interaction with the dendritic cells. And so it would also be very interesting to see how the microbes in your work that you just talked about would affect the different subsets of CD4 T cells, as well as the license to kill signal giving to the CD8 T cells. Now, as a transition to Marieke Fransen, the last talk, Marieke also has explored local immunotherapy. And in fact, when she still was in my lab, she was investigating the effect of local immune checkpoint blockers versus systemic. And what you see, if you have a systemic one on the top line, which is a high dose and leading to high serum levels of immune checkpoint monoclonal antibody, which was very toxic, in particular to the liver. But it did induce circulating tumor-specific T cells. But the same effect could be reached with local low-dose, slow-release antibody administration in the vicinity of tumor-draining lymph nodes. And that was associated with low serum levels and no appreciable toxicity to the liver. So by all accounts, I think that the delivery of checkpoint blockers can be much less toxic at much lower doses used when local therapy is applied. And this comes down to the same principles that, Laurence, that you have used in investigating the microbiome, which is basically also a very local effect, but with enormous systemic consequences. And so, without further ado, I just want to say that we are very proud of you. And in these two pictures that you get from the internet. And so, a great pleasure to see you over the years and interact with you. Thank you.
Thank you very much, Kees, and thank you to the organization for allowing me to participate in this wonderful event in honor of Professor Zitvogel, and my warm congratulations. I would like to continue a little bit about what Kees just touched upon and explain a little bit more. So I now work in Amsterdam, but before I worked in the lab of Kees in the LUMC, and some of the work I will show is still from there. So I can shorten my introduction. Immunotherapy with checkpoint blocking antibody is clinically approved in many types of cancer. It's very effective, however, only effective in a small part of patients. One of the major problems, though, is that there are some really serious immune-related side effects, and that the costs of these treatments is very high, not just due to the treatment being expensive, but also to alleviate the side effects. these patients often have to be admitted to hospital for their side effects. So we wondered if maybe we could do something about this, and we came up with the local administration strategy. What we did was we treated tumor-bearing mice with a low dose of CTLA-4 blocking antibody in a slow-release agent, so an oil and water emulsion, close to the tumor and close to the tumor-draining lymph node. And we compared it to the standard high-dose systemic treatment. And if you see here in the survival plot, if you don't treat, most of the mice succumb to the tumor. If you treat with a high-dose systemic injected anti-CTLA-4, a lot of the mice recover because they can clear the tumor. But if you treat with a much lower dose of anti-CTLA-4 close to the tumor, this is an 8 times lower dose, then the effect is almost the same as the high systemic dose, and a lot of these mice can clear the tumor and have a good anti-tumor response. This has to be a locally applied dose, because if you apply the same dose on the opposite flank, so away from the tumor, the treatment does not work at all. And this is important. When we showed this data to people, we always got the question, that's all nice for a mouse with one tumor, but patients often have metastases or can have metastases. What are you going to do? So what we wanted to know was, does our treatment elicit a systemic immune response? So we analyzed in these mice treated with the high or the low dose anti-CTLA-4. We analyzed the anti-tumor T-cell response in spleen and in blood with tetramers, and we found that the local low-dose treatment of anti-CTLA-4 gave just as many activated anti-tumor T-cell response in spleen and in blood as did the systemic high-dose treatment. Then we wanted to know, are these T cells also functional? So we used this system where we had mice with 2 tumors, a somewhat bigger tumor on one flank, which we call the primary tumor, and a little, somewhat smaller tumor on the other side, which we call the secondary tumor. And we gave these mice a low-dose local injection close to the primary tumor, but then we measured the outgrowth of the secondary tumor. And as you can see here, the treated mice, the secondary tumor did not grow out. As a control, we had mice that did not have a primary tumor, but only had a secondary tumor. And if you treat them with a low dose, then the tumor does grow out. So the low dose local administration elicits an anti-tumor T-cell response, which is capable of clearing the secondary tumor. And when we checked the amount of therapeutic antibody in the serum, and as Kees just mentioned, this is indicative for the amount of side effects, as is also seen in patients. Due to the fact that we used a slow-release agent to inject these low-dose anti-CTLA-4, we could detect up to 1,000 times lower anti-CTLA-4 blocking antibody in the serum of these mice. Now, this led us to think, by the way, we showed this with anti-CD40 agonist antibody, with CTLA-4 blocking antibody, and we also showed this with PD-1 blocking antibody. In all cases, a low dose applied locally is as effective as a high dose systemic. Now, we wanted to know, what are the local factors that make this happen? Now, everybody has their own favorite cell kind molecules, and I now realize I should have added the gut microbiome to this list. The vast majority of scientists was all focusing on the tumor microenvironment, but we decided to focus on the tumor-draining lymph nodes, because draining lymph nodes are the orchestrators of immune responses, and we were convinced that they played an important role. So we set up this experimental design to analyze the involvement of the draining lymph nodes. So we had mice, we injected tumor cells. When the tumors had formed into a small solid tumor, we surgically resected the tumor-draining lymph nodes, immediately followed by PD-1 blocking antibody therapy, and then we followed whether the tumor grew out. And this is example, the tumor is located pretty much here, and we resected the inguinal lymph node and the axillary lymph node. And don't worry, we didn't open the mouse up like that. This is just for an example. As a control, we did do mock surgery where we made a small incision but leave the lymph nodes undisturbed, stitched the incision back up, and they were our controls. So what you can see, if mice that still have their lymph nodes, they don't get treated. All the mice succumb to tumor burden. If you treat them with PD-1 blocking antibody, most of the mice are able to clear the tumor. But if you resect the lymph nodes right before giving PD-1 blocking antibody, then this clinical efficacy is strongly reduced. There is still some effect of PD-1 blocking antibody, but it's very much reduced compared to the mice where the lymph nodes were in place. We saw this when we gave local application of PD-1 blocking antibody, but we also saw this when we gave a systemic high-dose application of PD-1 blocking antibody. We also saw it when we gave PD-L1 blocking antibody. So in all those cases, the tumor-draining lymph node is actively involved in orchestrating the anti-tumor T-cell response. So we wanted to know a little bit more about that, and we looked into the tumors of these mice. What is going on after this treatment? So if you don't do anything, about 30% of the cells in the tumor is composed of immune cells. If you treat with PD-1, that stays pretty much the same, but if you resect the tumor-draining lymph nodes, then the amount of immune cells in the tumor is drastically reduced. So it seems like there is an active migration back and forth between the tumor and the lymph node all the time. And now if we look specifically at the CD8 T cells in these mice, treatment with PD-1 makes the amount of CD8 T cells in the tumor increase, but if the tumor-draining lymph node has been resected, then that does not happen. And we try to prove this in another way as well. So we treated tumor-bearing mice with PD-1, and in this group we also treated these mice with FTY720, which is an inhibitor for S1P receptor, and it effectively locks T cells in the lymph node. So T cells that are in the lymph node cannot go out. And what you see then is that the effect of PD-1 blocking is abrogated when T cells cannot leave the lymph node. So this shows the important role of the tumor-draining lymph node in activating the anti-tumor T-cell response. This was published in 2018, and after that I moved to Amsterdam in order to study this further in patient samples. I joined forces with Professor Tanja de Grel, who had been pioneering translational research and clinical trials of local administration of immunotherapy, and who shares my interest in the role of the tumor-draining lymph node. So that's how I got to be involved in some of the translational analysis of her trial of local low-dose CTLA-4 blocking antibody in melanoma patients. In this trial, early-stage melanoma patients first had their primary tumor resected, but the tumor-draining lymph node was left in place. A very low dose of tremelimumab was injected in the scar of the tumor resection, and the tumor-draining lymph nodes were resected 1 week later. This work was published in Science Immunology this summer, and it shows a lot of the translational analysis that were done on immune cell activation within the tumor-draining lymph node, but also in the peripheral blood. And one of the examples I will show you here, this is an ELISpot analysis of T-cell responses in blood against NY-ESO and MART1, which are melanoma antigens. And what you can see is that a low dose of anti-CTLA-4 blocking antibody targeted to the tumor-draining lymph node leads to a strong increase of anti-tumor T-cell responses. Unfortunately, there were a few patients that did not respond, but the majority of patients either augmented the response that was already present before treatment or
developed a de novo response. And one of the other studies that I'm now working on is in non-small cell lung cancer patients. So I'm currently working on translational research in the INCREASE trial, which is a trial where locally advanced non-small cell lung cancer patients are treated with neoadjuvant chemoradiotherapy, which is the standard treatment for these patients. But the INCREASE trial adds double immunotherapy to it. So CTLA-4 blocking antibody and PD-1 blocking antibody before scheduled surgery. And because of this, we have the possibility to study all the tumor draining lymph nodes and the tumor and the blood for our scientific interest. So what you can see here is a staining of tumor draining lymph node material from an INCREASE patient and from a matched control who received neoadjuvant chemoradiotherapy but no immunotherapy. This is a staining of CD8 T cells in brown and Ki67 in pink. So what you can see is that the INCREASE patient has a strong increase in activated CD8 T cells and probably a lot of other immune cells as well. This is a summary of all the INCREASE patients' tumor draining lymph nodes compared to matched controls. And there's a clear increase, which that's a very appropriate name for the study, of activated T cells by this treatment. And if we look at correlating peripheral blood samples, we also see a strong increase between baseline and after treatment of these activated CD8 T cells. We are still working on this, and we hope to have a lot more analyses of these tumor draining lymph nodes over the coming years. So this work has been done partly in the LUMC in Leiden and partly in Amsterdam. I should have one of those nice slides that you also had, Professor Zitvogel, with lots and lots of pictures of all the people. Unfortunately, I don't have one, but these are at least, oh, I'm so sorry. Oh, I can't go back, can I? These are at least, let's say, the usual suspects. Thank you very much. All the speakers and also Laurence Zitvogel to join me for a roundup of this day. And it's your last chance to ask a question. And of course, I'm looking to Bob Pinedo first. Maybe you have a first question? No? Okay, then I give the floor to all of you. I can imagine so many different topics. You have a question. And please also mention your name. Yes. Ah, yeah. That's okay.
My name is Mark Peters, oncologist at Antwerp University in Belgium. I have a question and congratulations for all the information, especially to Laurence. The question is, the microbiome is playing a role not only in cancer, but in different diseases. And we need to focus on the patients that have benefit of influencing the microbiome. How do you see the development of personalized medicine? Meaning, how do we tackle the development of biomarkers in the future? Will it be cancer-related, therapy-related, disease-related? How do we see the development? Because this is important. You said we cannot offer all patients diet. We cannot offer all patients FMT. So personalized medicine will be important.
Can start. So this is a very broad question and very, let's say, anticipating and visionary question. So we have anticipated this problematic as well by launching what we call Prevalung. I think we launched that with Lex when he was still there. It was a study aimed at focusing on cardiovascular diseased heavy smokers. And we were wondering whether the biomarkers that we are currently using can actually predict which one of these patients that were inflamed but cured from their cardiovascular disease, and they were more or less stopping their smoking habit, which one would develop the lung carcinoma or another tobacco-associated carcinogenesis. And so we were surprised. So we looked at all the biomarkers that most of you are currently investigating. So immunomics, proteomics, inflammomics, metagenomics, metabolomics, barrier fitness, etc. So it's 7. That's why I had an octopus on my slide, because each arm of the octopus was actually one dynamic of biomarker. And to make it short, we discovered very surprisingly that, so we got 17 lung cancer out of 508 cardiovascular heavy smokers, so 2% yearly incidence of lung carcinogenesis. And when we looked at the biomarkers, so in all except 3 persons, all the biomarkers were reshuffled, so meaning they were completely disabled, and they were harboring a systemic disease, as if everything was connected. So, and one of the most, let's say, presumably most sensitive marker was the SHIP, the clonal hematopoiesis of the bone marrow, the senescence of the bone marrow and the mutations. So these mutations that are like JAK2, TET2, DNM3TA, etc., were associated with inflammatory behavior of the monocyte, IL-1 beta production, and increased systemic inflammation. So it goes back to the notion that once you have an inflammatory disease, it's not just the organ that is targeted that is a problem. There is a general systemic problem where everything is interconnected. What comes first remains an open conundrum. But basically, there is no, I think it's going to be difficult to personalize, except if we longitudinally follow up the patient, as done actually at the Weizmann Institute. They call that cohort in a box. So every single person, 35-year-old person, should be screened from everywhere, followed up every other 5 years, and then we would record, mental disease, Alzheimer, arthritis, and so on, malignancies, cardiovascular, and we would be able, presumably, to conclude that maybe there is a ranking and or a chronological order of the diseased organ or disease compartment. But to respond to you, I think it's a crucial problematic that we need to prophylactically intervene on the cause of the general systemic inflammation and decompensation, because if we don't do so, of course, we will have, different type of disease, and I think that cancer is just one amplification, of a chronic inflammatory disorder or a decompensated senescence.
Yeah, I totally agree that prevention of disease is, of course, much better than trying to cure late-stage disease. But we are, of course, still confronted with many patients with overt cancer, even metastatic cancer. And I personally, I want to say something about my personal experience in the last half year. I happen to have two friends of my age who happen to have neuroendocrine cancer with liver metastasis. And I found out that for neuroendocrine cancer, it's not routine to have the cancer genome sequenced. And yet, I insisted on having this done. And what then came out is that in both cases, actually, there were other medicines possible that are available in the pharmacy that were repurposed. And in the Netherlands, there is even an organization called DRUP, D-R-U-P, which on the basis of, let's say, sequencing by the Hartwig Foundation, which is a not-for-profit foundation, which is basically a charity. But I find that all patients with cancers should have this DNA sequencing, because not only were they now offered alternative treatment that only came out of the DNA sequencing, but they also are now susceptible or open to immunotherapy treatment that otherwise would not have been possible, including personalized vaccines against neoepitopes that are unique for each patient based on the unique mutations of that particular, including also driver mutations, which are particularly attractive to target with both small molecules and cancer vaccines.
And would you also conclude that sequencing gives us now more information than checking someone's microbiome?
Well, I think Laurence is absolutely right. You need to sequence the microbiomes and pick out the right ones and transplant them and use them, but also be aware of what that specific disease of that patient is. Yeah. So we have all the tools to look at that.
Yeah. Other question?
Yeah. May I add one comment?
Yeah, one comment, and then a question.
We know that the sequencing programs have been disappointing in the sense of the absolute percentage of patients who benefit from all this sequencing. So it's incidental. And at the same time, anti-PD-1 was discovered, and we had the biggest blockbuster in the history of cancer medicine, meaning that we also have to deal with the first 10 laws of Moses. And we know only 2, and that is anti-CTLA-4, it's anti-PD-1 in that area. And so when we look about microbiome, one of the first questions that we actually should address is, what is the common denominator, first law of Moses, in the microbiome that is representative of a chronic inflammatory status that may be common to cancer, Alzheimer, multiple sclerosis, and morbid obesity? Is that the case? What are the overlapping circles, and what is the central or core circle, and what can we detect from that? Because then at least we could concentrate our efforts to find out how to deal with that core microbiome, and may have multiple impacts on multiple chronic diseases. What do you think? Because you know this.
Yeah, exactly.
Then we go to the question.
So I would like to highlight that Wiersma, Rins Wiersma in the Netherlands, has published this beautiful Nature paper a couple of months ago in April, analyzing more than 8,000 DNA from families, Dutch families. And at the end of the road, he concluded exactly what you just said, that there is a group of bacteria that is dominating the scenario, and a group of functional genes encoding a disbalance between the functionality, so the metabolism of these gene functions. And in fact, for me, the way I read it is that it's belonging to this enterocluster genus that I was talking about. And, but it's now that we know that, and it's common to all inflammatory chronic disorders. I disagree with your introduction, by the way, because I think that it's not because each single study is not showing the same particular species or strain, because there are thousands of strains. But if you project, and I didn't have the time to do that, but if you project the metagenome into what we call ecological system, topological ecological system, it's like a zoo where we would have cages of lions, of birds, of whatever. So these cages, when they are open, it's a mess, of course, and you have of course, a reshuffling that is usually deleterious. But if now you look at the cages of species rather than the single guys in one single cages, then it's completely clear. The picture is completely clear. So yes, the enterocluster genus is I don't know if it's the cause, but it's highly associated with this inflammatory disorder. But the question we have is how to get rid of that. So that's then.
Well, now Jessica, you can respond, of course. And then I go to your question.
Well, it was meant as a little timeline at the time because that was all they knew before they started the first 2 clinical trials. They didn't know what was published recently, and there's definitely a lot more information at the moment about which parts of the microbiome might be associated. You showed a very nice picture of across tumor types what was associated with or without response. But still the interesting thing is, like, in one of the 2 clinical trials, it was the picture on the left, maybe for the people who paid close attention to the figure, there were 2 donors used, one donor with a complete, the 2 donors were both responding patients, so melanoma patients who responded well, donated features, but only patients who received the FMT from one of the 2 donors responded. So just the fact that one patient is a responder doesn't mean that the microbiome might be good enough to also give the recipients of the FMT a response to their immunotherapy. So that's why there are still, lots of things unknown. So just the fact that a patient may have a really beneficial microbiome is not enough, or a patient might be responding despite the fact that his microbiome is not ideal because he has a high tumor mutational burden, or he has a high number of tumor-infiltrating lymphocytes in the tumor. So that's why we also want to do this study.
Okay. I'm really curious to find out what your question is, and also mention your name.
Yes, I'm Rick Scheper from the VU Academic Medical Center here in town, and I'd like to get back to the low-dose immunoregulation, let's say the opportunities for low-dose immunoregulation, and it connects also to the work of Laurence Zitvogel, which I've studied in several times in many years. Anyhow, around, I think 25 years ago, with a student, Gijs Boerichter, who later came to work with the Dutch Cancer Society, we discovered that taking a very low dose, a non or a marginally toxic dose of cytostatic drugs, alkylating drugs, but also etoposide, anthracyclines, whatever, but not the anti-metabolics like 5-FU or methotrexate, but all those drugs, when you gave them at a marginally toxic dose at the site of antigen stimulation, then the lymph nodes were at least twice or thrice, 3 times as big as they used to be. So basically we did it as part of studying contact allergy. We wanted to induce tolerance, which we did not get. No, instead we got an enormously exaggerated T-cell mediated immune response. But in the 90s, we did not have the reagents. We published 2 theses, 10 papers in nice journals and whatever. You can all find them back. But as far, so
the question
then
is?
cytokines, and everybody forgot about it. So do you know, has anyone followed this up? Do you think about cytostatic?
Yes or no?
Well, you were way ahead of your time. But I believe that very often for immunotherapy of cancer, we are overdosing the checkpoint blockers and we are overdosing the chemotherapy. So that's what your studies likely also showed, because what we found in our studies that immunosuppressive myeloid cells, which are rapidly dividing cells, are much more sensitive to the most chemotherapeutics than the lymphocytes. Particularly the T lymphocytes were completely untouched by those low-dose and even high-dose chemotherapeutics, and therefore they can perform much better.
Marieke, last words, maybe?
I actually think maybe we should pick that back up. Now, is it possible, do you know if those very low doses actually did kill some of the tumor cells? That I would be interested to know that. Or was it really just about taking away some of the immune suppressive cells?
We at the time it took away the suppressor T cells and the suppressor cells, later called the regulatory T cell. But we did not have the reagents. We studied it in the line 10 guinea pig model, and it also worked perfect there. But we did not have the reagents, and we have not seen killing of the tumor cells. but the size of the tumors, of course, was
yeah. Yeah, you would want to find the sweet spot where you do get some tumor cells killed and the immunosuppressive cells taken out, but not everything. Because I think that's the problem with the high dose.
So, yeah, final words.
So, you know that adjuvant anti-PD-1 therapy in lymph node positive melanoma patients is standard of care. That is 18 doses of anti-PD-1 after lymph node clearance. By simply giving the first 3 doses of those 18 doses prior to the lymph node surgery, you get a 42% reduction of relapses. So neoadjuvant therapy is completely changing and will change the management of multiple tumors. It starts always with melanoma, with the combination of anti-CTLA-4 plus anti-PD-1 for palpable nodal disease, which will fail for 80% within the first 18 months after surgery. You get 60% pathologic complete responses, and those patients are cured because we've seen only 1 relapse. This is the work of Christian Blanc here at the NKI. 1 relapse in 30 months. We have never ever seen such data in melanoma management. The same thing is now rolled out in cutaneous squamous cell cancer in the face. The surgery is going to disappear. And within 5 years, sentinel node staging will disappear because we will give immunotherapy at early stage at low doses. That's what's going to happen. We'll just have to do the trials.
And what, in your opinion, is the role of the local draining lymph nodes in that?
Well, that's because they are still there. You shouldn't take them out. That's the whole point. This is priming forces. And what are we doing with surgery, taking out the priming forces now that we have anti-CTLA-4 and anti-PD-1? Before, you had to take them out for local control. Now, you should give and work with the tumor cell containing lymph nodes to program the whole system. And so all those early signs were already there. This is now going to dominate the whole works, neoadjuvant immunotherapy, not just in melanoma, not just in cutaneous squamous cell carcinoma. Not a single patient with an MSI rectal cancer should be programmed anymore for rectal cancer surgery. They should all be referred to the few centers who give anti-CTLA-4 plus anti-PD-1 up front. The 92% pathologic complete or near complete response rate means they don't need rectal resections anymore, and actually they will be cured. But this is only 5% of colorectal cancers, which, by the way, is much more than all the melanoma patients in the world.
Thank you. That having said, I would like to thank the panel. Thank you all speakers today, and of course, Laurence Zitvogel. I'd like to thank all of you for being here. I'd like to thank everyone watching online, wherever you are in the world. I'd like to invite you to the reception, and thank you for being here.